presentarles a nuestra invitada del día de hoy, la doctora Mireia Valles-Colomer, que nos acompaña de Barcelona, España, de la Universidad Pompeu Fabra. La doctora Valles-Colomer tiene una licenciatura en biotecnología, perdón, en España, que obtuvo en el 2012. Después se fue a hacer una maestría en ciencias de biología molecular en la en Bélgica. En el 2014 inició su doctorado, el cual concluyó en el 2019 en la Universidad de Lovaina, en Bélgica. Tanto la maestría como el doctorado ya los hizo en el campo que ella se especializa en el análisis de la microbiota intestinal. Y después continuó con una estancia postdoctoral con el doctor Jeroen Raes, también trabajando con microbiota en la Universidad de Lovaina, en Bélgica. Su segundo postdoctoral lo realizó en la Universidad de Trento, en Italia, donde con el doctor Nicola Segata continuó trabajando en la transmisión de la microbiota oral y del intestino, para que finalmente en el 2023, hace un año, el año pasado, le dieron la oportunidad de tener su propio grupo en la Universidad de Pompeu Fabra en España, donde evidentemente pues continúa trabajando en ese tema tan interesante que es la microbiota intestinal y bueno, en general la microbiota en mamíferos y con un enfoque especial en humanos. La doctora Valles-Colomer solamente tiene 37 publicaciones, pero les quiero decir que tiene ya más de 9,000 citas a su trabajo, y como podrán ver, bueno, pues sus trabajos han sido publicados en unas de las revistas más relevantes del área de la biología celular, de la biología molecular, y siempre trabajando con microbiomas. Entonces, realmente el impacto que ha tenido la investigación de la doctora Colomer es bastante impresionante, como van a ver también de su presentación. Bueno, la doctora Colomer ha participado en su número de cursos como profesora, en un número muy grande también de pláticas en diferentes simposios y congresos alrededor del mundo. Tiene varios premios, del cual me gustaría resaltar nada más el premio de los F1000, o sea, el 1000F, que reconoce los artículos que, de acuerdo a esta grupo o asociación de investigadores, consideran que van a ser de los más relevantes en el campo, y se lo otorgaron por su artículo publicado en Nature Microbiology en el 2019. También participa como revisor en diferentes revistas, evaluador en diferentes fundaciones para financiamiento de la investigación, en muchos comités de bases de datos, entre otras actividades. Evidentemente, pues también ha dirigido tesis de doctorado que ya concluyeron y que están en progreso, tesis de maestría y también alumnos de licenciatura. Entonces, realmente no me queda más que agradecerle a Mireia que haya aceptado la invitación para platicarnos el día de hoy de su investigación que realiza en Barcelona. Adelante, Mireia, por favor.
Gracias. Muchísimas gracias por esta introducción. Pues sí, estoy encantada de estar aquí hoy, también después de la charla muy interesante que tuvimos ayer con los estudiantes. Voy a hacer mi presentación en inglés, que la preparé así. Me dijeron que no había preferencia, pero luego para las preguntas, pues en lengua que sea, no hay problema. ¿Me podéis confirmar que se está viendo la pantalla de presentación?
Se ve bien y se escucha bien.
Perfecto, genial. Pues voy a empezar. So yes, I'm going to present an overview on our research on the neuroactive potential of the gut microbiome and its social transmission. So this is what I've been studying during my career. So first, I'm trying to understand how the microbiome could play a role in our mental health, and then also understanding how we actually acquire these microorganisms and what role they could play on our health. Okay. So indeed, we study the human microbiome, which we define as the collection of microorganisms that reside in and on the human body and their theater of activity. I don't think at this point we need to convince people that the microbiome is important, but we have as many bacterial cells as human ones, roughly, in our bodies. So some years ago, it was said that we had a lot more bacterial ones, but actually now we know that it's not that much, but still more or less half and half. So there are a lot of them, and also we have many more bacterial genes than human ones. They reach their highest densities in our intestines. So on average, we have 10 to 10 bacterial cells per gram of feces. And they definitely play a role in maintaining host health. So we have a lot of associative studies in which alterations in the composition of the microbiome are linked to many of them. And you can see that this list of diseases does not only include gastrointestinal diseases, but also many conditions that we would think traditionally as linked to mental health, like depression, Alzheimer's, or multiple sclerosis. So I'm leading, as it was introduced, the microbiome research group at the Medicine and Life Science Department of the University Pompeu Fabra in Barcelona. And we have these two main lines of research. One is studying the microbiome gut-brain axis with computational tools, and the other is trying to understand the transmission landscape of the microbiome. So could the microbiome play a role in mental health? This is a question that can be seen as something quite far-fetched, but actually we can see that we have evidence from that already for quite a while. So one of the first instances in the literature on an interaction between the gastrointestinal system and brain health was already presented in the late 19th century when visceral stirrings were proposed as part of the emotion of fear. Then we also know for a long time that human neurotransmitters can affect host-microbe interaction. The first example was adrenaline making bacteria more infective. And also that microorganisms can actually synthesize many of what we consider human neurotransmitters. The first example was acetylcholine by Lactobacillus plantarum, but now we know that most neurotransmitters can actually already be produced by the bacteria. So this field has been expanding a lot in the last decade. We can see really that it was, although we have instances from a lot before, from 2010 or so, we got this exponential growth. And we define the microbiota gut-brain axis as a bidirectional communication system integrating neural, endocrine, and immune signaling between the gut and the brain. And what I find most interesting is that members of the microbiome can not only influence these neural, endocrine, and immune communication routes, but also they directly can produce and degrade many neuroactive compounds. So these include the neurotransmitters that I mentioned before, but also short-chain fatty acids, like butyrate, hormones like cortisol, the main stress hormone, immune modulators, so on. So the way we study the microbiota is we get samples for the gut microbiome. It's typically fecal samples. These are non-invasive, easy to obtain. We perform DNA extraction of the whole community. And then traditionally, there are these two approaches. One is marker gene sequencing. Typically, the 16S ribosomal RNA gene was selected. But this, as we sequence only one gene or a fraction of it, we get all information on the taxonomic composition of the sample. So we are moving more and more towards shotgun sequencing, also as the costs now are quite similar, which gives us information on both taxonomic and the functional composition of the sample. So who's there, but also what can they do? So we just go, we get this sample, we extract the whole DNA, and we sequence everything that is out there. The issue here is that interpretation of especially the functional data is really far from straightforward. So our first approach was developing an approach to translate this data into the neuroactive potential of the microbiota. So from all these sequencing reads, trying to see what are the functions that the microbiome could encode and that could actually influence our health. I'm not going to get too much into detail here, but we define this as a set of gut-brain modules, which define steps, so relevant steps of the microbial metabolism of neuroactive compounds. So we have just one example on the right, so which would be so the process through which bacteria get from dopamine to DOPA, which is also a relevant neuroactive compound. So then we built all of these, we annotated based on metabolic pathways when they're involved in different diseases based on the literature, and also their potential of both crossing the intestinal epithelium and the blood-brain barrier. And then we tried to validate it based on experimental evidence. This computational approach led us to build the first catalog of neuroactivity of the gut microbiome. At that point, we could see that there are some functions that are basically ubiquitous in bacteria, so most of them just have them, whereas there are some others that were quite rare. These could be very important functions, but still they are only coded by a small fraction of the bacterial genomes that are typically there in our intestines. And it was interesting for us to see that for many of these rare functions, they were actually encoded by bacteria that are known as pathogens or opportunistic pathogens. And they had already been reported to respond to gut neurotransmitters. So now we know that besides responding to them, they can also directly produce or degrade them. We went one step further and we compared then this distribution of these functions in this catalog of gut genomes to bacteria isolated from the environment. So free living organisms from, for example, soil or water environments. And we saw 13 of them were strongly host associated, suggesting that these functions would be relevant for bacteria to interact with us. These functions included, for example, all three pathways known at the time to synthesize the neurotransmitter GABA in microorganisms. So that was something quite relevant for us, knowing that GABA synthesis could be beneficial for these bacteria, and then it could actually interact with our bodies. Then we also assessed its distribution phylogenetically, and we saw that these are quite widespread. There is not really like one phylum or one genus in bacteria that is a winner, so has a lot more of these functions. But still, they were quite rarely encoded in plasmids, suggesting the difficulty of horizontal gene transfer of these complex traits. So after having this idea, having this catalog on the distribution of these functions in the diversity of bacteria that was known at that point, we went into the real samples of our population. So to assess the association between these functions and quality of life and depression. We did this in the Flemish Gut Flora Project dataset. I was working in Belgium at that time, and we had access to this cohort with more than 1,000 individuals, from which we had fecal samples and also very detailed information. So on quality of life, we had the RAN, Health-Related Quality of Life Questionnaire, that has some mental health and some physical health indicators. So we could see this way what are the microbiome traits that were associated with mental and physical health indicators. And then we also had a diagnosis of depression based on the participants' general practitioners. So we had in our cohort 121 individuals with diagnosis of depression, a bit less than half being on antidepressants. So first that we explored was the association between quality of life and depression indicators and the taxonomic composition of the microbiota, so the abundance of a specific bacteria. We saw that quality of life and depression indicators play a smaller role as compared to the main known covariates of microbiome composition, so anthropometrics, where we had age, gender, and body mass index, or gastrointestinal parameters, where it had gastrointestinal diseases, and also Bristol stool score, which is a scale that goes from 1 to 7 based on stool consistency, and that we can take as a proxy for gastrointestinal transit time. So yes, quality of life and depression scores were not as important for microbiome composition as compared to these variables. But still, when we built a cumulative model, which is what we see here, we could see that the fraction of inter-individual variation in microbiome diversity that they explain is still non-overlapping. So they add something extra besides anthropometric and gastrointestinal parameters. So it had body pain, physical functioning, general health. So 3 of the quality of life scores, and also then diagnosis of depression that entered this model. Then next step was looking for specific associations between quality of life scores and diagnosis of depression. We found significant associations after correcting for multiple testing and partialing out the influence of these anthropometric and gastrointestinal parameters, as they could be confounders for our analysis. And we saw that there were mostly 2 bacteria, Faecalibacterium and Coprococcus. So these 2 genera were positively associated with both mental and physical quality of life scores, whereas Flavonifractor was negatively associated. This was quite making sense based on the literature, because Faecalibacterium, Coprococcus, they produce butyrate, which has anti-inflammatory properties, and is considered beneficial for our gut health, whereas Flavonifractor already has some reports of associations with different diseases. Here, the bubbles that you can see that are labeled with a tick were the associations that were validated in an independent data set. So this was the Limefline's DEEP from the Netherlands, which was a similar cohort to the Flemish Gut Flora project, with also around 1,000 of individuals and similar data that we got from them. So this, we see them as the strongest associations and also the main follow-up targets for next research. Then for depression, we see that actually it was quite overlapping with quality of life associations. And we see that Coprococcus again, and then Dialister were in reduced abundances in patients with depression as compared to the healthy controls. And this association was also validated in the independent cohort. And was also still standing when taking into account medication, because it could be that medication is what is influencing the composition of the microbiome. For many drugs, there is clear evidence that those could influence. But we see that in this case, and differently from these other 2 associations, even when taking into account the influence of medications, these still stay. And actually, these 2 were already reported in the literature based on smaller case control studies. So then we applied the framework that we newly developed. And indeed, we found some associations, mostly with GABA pathways. And then the strongest one was DOPAC synthesis, the dopamine metabolite that I mentioned earlier, and that has altered abundances, for example, in patients with Parkinson's disease. So we saw that mental scores, mental quality of life scores, were positively associated with DOPAC. And one of the associations, the one with the social functioning score, was validated in the independent data set. So when this was published 5 years ago now, there was a lot of talking in the field, thinking that we were in the early days, but we might be at the beginning of a new revolution in how we understand and possibly treat depression through targeting the gut microbiota. It's been for many years that there is no new drug for depression in the market. So we think that targeting the microbiota might help at least a subset of the patients. So now we keep developing our computational approach. We are extending the number of pathways, but also the number of genomes in which we are testing it, and we are updating the whole approach to make it faster and also more systematic and more solid. But overall, in the field now, the consensus, based also on other studies that have become available, is that in anxiety and depression, the main signatures at the microbiome level is that there is decreased relative abundances of short-chain fatty acid producing bacteria, the fecal bacterium and coprococcus, whereas taxa that are associated with inflammation are in increased abundances. So this leads to propose this pathway in which in health, we just have a health-associated microbiome that is keeping homeostasis, whereas in an inflammatory state, there is advantage for these bacteria that help causing inflammation. Then there is more dislocation of these bacteria into circulation, and it just goes into negative feedback loop that is just sustaining inflammation. So from this, then we went on, and the COVID-19 pandemic started. And we started doing a project in Spain in which we're trying to see which are how was the microbiome changing during infection, and if there was any association with depressive, anxiety, or post-traumatic stress disorder symptoms. We saw that indeed COVID-19 infection affected microbiome composition. There have been many other studies on the subject now, and although they do not all agree in the specific alterations, there seems to be some alteration in general. But what we started thinking is, what about isolation? So it seemed that when having less contact with our social network, this would also influence our microbiome. So we then started studying, and that's when I moved to Italy, the gut microbiome with social networks. So that's an interesting question, in my opinion, as a lot of the research efforts in the last 10 years or so have focused on understanding which are the factors that influence the composition of our microbiome, such as we know now that diet, genetics, lifestyle, delivery mode, medication, and so on, all have strong effects on our microbiome. But there is just this missing question, which is how do we actually get these bacteria and how do we transmit them to others? And it has important implications, for example, for the non-communicable diseases like cancer, obesity, or diabetes that are associated with alterations in the microbiome. And by definition, if these diseases are associated with alterations in the microbiome and the microbiome is transmissible, then these diseases could have a transmissible component. So maybe we should rethink whether they are actually communicable. So we think indeed this plays a strong role. It's important for our health, and it's a very fundamental question that has not been addressed much. So the way we study this time is, instead of trying to get in the functional composition, is pushing the boundaries on what we can do with the taxonomic composition of the microbiome. So we try to analyze the microbiome not only at the species level, but at the genome level to do a strain level analysis. So this way we can build trees like the one you can see here, which is similar to what is used in epidemiology for single microorganisms, to see and map the transmission of specific bacteria in populations. So what do we know? The first way of transmission that has been studied is the acquisition of the microbiome in early life. We have now evidence of mother to infant transmission. So when we are born, we get most of our gut microbiomes from our mothers. And then this is our first inoculum, which is followed by microbial succession processes. When this was published, it was also observed that maternal strains are more persistent in the baby gut than others, environmental strains from other sources, suggesting that they are better adapted. But we then wanted to go one step further. And what we did was assessing the transmission of the microbiome across multiple generations. So seeing if it's possible to get your microbiome not only from your mother, but for example, from your grandma as well. So we enrolled 24 families in Belgium. I did this project there still, with 3 to 5 generations of women. At that point, we managed to profile 51 different bacterial species at the strain level. And what we saw is that this is something that does not occur very frequently. So there is a lot of transmission from the mother to the daughter, but not so much from the grandma that goes through each generation. But in some cases, we did see examples of that. So for example, this would be one. So for Alistipes under donkey, and indeed, it's you can see here that most of the bacteria for which we observe these are from Alistipes or Bacteroides bacteria. We observe this. But then when we went into a bit into more detail and we were trying to see whether this was actually due to kinship, so the grandma passing the bacteria to the mother and the mother to the daughter, we saw that the cohabitation was even playing a stronger role, suggesting that this could actually also be horizontal transmission. And we think this makes sense because infants have a much reduced microbial diversity, whereas adults have a lot higher. So it's not possible that we get everything from our mothers when we are born. So what do we know on horizontal microbial transmission? So first of all, it's known for quite a long time that people that live together have more similar microbiome profiles. But this could be, of course, because people have a similar diet or follow similar lifestyles. But the first indication of horizontal transmission of the microbiome, so finding the same genome of in different individuals, so in households, was suggested by a study in the Fiji Islands that showed transmission within households and also notably within spouses. So this is something that is interesting, especially because many bacteria in our gut microbiome are anaerobic, so they cannot survive outside our bodies and in the presence of oxygen. Except for some that have evolved survival mechanisms, for example, spores. So we did a large-scale study on microbiome transmission where we analyzed almost 10,000 samples from different data sets around the world. As I said yesterday, we don't have Mexico yet in our map, but we hope to have it soon. And yeah, we got samples that are publicly available, but also others that we collected through other collaboration with local partners. And we did this for the gut and for the oral microbiomes, so with fecal and saliva samples. And we did metagenome profiling, so assessing strain transmission with an approach based on metagenomic assembly, which let us also profile transmission of bacteria that have never been cultivated. So this way, through genomics-based analysis, we can also track the transmission of bacteria that we cannot really study in the lab. Our first point was going for a systematic definition of strain, because yeah, we are comparing genomes, but how close should two genomes be for us to consider that this is equal, so similar enough so that it was transmitted? It could be that if our parameters are not strict enough, we just consider transmission when there was not. Or if we are too strict, then if within one individual there is mutations accumulating of one bacterium, then we just consider it something different, so that this bacterium was lost, where it just accumulated some mutations. So went through some methodological development approaches to see where we should set the thresholds for different species. And we think it's important to set different thresholds for different species because different bacteria evolve at different rates in our gut. So this is a process that we have implemented in publicly available software. We also have tutorials available. And then the next step was also saying, okay, it could be that transmission is happening directly, so from one person to the other. But the alternative is that can also be acquisition from a shared source. So two people eating the same commercial food, but even if they are not in contact, then we see the exact same genome in them. So to get rid of these second cases, what we did was removing from our analysis all the bacterial strains that were too similar to the genomes of bacteria found in food. So this meant, for example, for Bifidobacterium animalis, which is typically found in yogurts, removing the whole tree. This meant, in this case, everyone had the exact same strain, the one in yogurt. Whereas for Staphylococcus thermophilus, as another example, it just meant removing some subtrees. So in this case, some people had the strain that is commercially available, also as probiotic in the pharmacies, but then there is also normal variation and diversity. So next step was assessing then with our thresholds how much transmission there is among individuals with different relationships among them. First of all, we saw that indeed, when we compared samples from the same individual, so my sample and my sample in 3 months, so we put a maximum of 6 months, and if there is no intervention, for example, I did not take antibiotics or I did not change my diet drastically, still, as a median, 87% of the microbiome remains there. Whereas with people that I never met in the same population or in different populations, there is a 0% of strain sharing. So these 2 points corroborate that our approach is solid enough. So even if there is someone else in the population that has a similar lifestyle than mine, there is 0 sharing. Whereas then we have in between samples, so comparisons between mothers and their offspring, people in the same household, or we also studied twins, and then other people in the same village. So first of all, mother to infant transmission. This is the most studied transmission route. We know indeed that we get most of our microbiomes from our mothers when we are born. There we see that in the first year of life, strain sharing with our mother is quite high. So half of the strains are shared and quite stable. So 50% during this first year. Then later on, when the infant acquires motor activities, it also aligns with weaning, then there is more, so here, bacteria acquired from other sources. It could be the environment or other individuals, and less sharing with the mothers. But still, we can see in this one data set that was from individuals in that are between 50 and 85 years of age in China, still with their mothers who are alive. They still shared a lot more with their mothers than with mothers of other individuals, suggesting that maternal strain sharing is first considerable, but also quite long-lasting. We also assessed then the influence of delivery mode, and we saw that vaginally delivered infants do share more strains with their mothers as compared to those born by C-section. But this strong effect is only seen until 1 year. So after 3 years of age, we do not see differences anymore. So there has been, this is something that has been debated a lot on the different specific studies that were published before. And what we see here is that, yeah, it's very important in the beginning, then not so much, but still there could be important neurodevelopmental windows that on which it's important to have this bacteria. And then while we had very different lifestyles in our cohorts, also with geographical spread, there was no significant effect of lifestyle after accounting for the age of the offspring. So next, we also studied whether other caregivers could also provide part of their microbiomes to the infant. We studied the father as the first choice of caregiver. And indeed, what we saw is that while the inoculum from the mother is very strong at first and then it decreases, that from the father starts lower, but then it's stable. Meaning that then by 1 year of age, we did not see anymore that the mother is contributing more than the father. So we can see that the mother at first has this very strong role, but then not so much anymore. Indeed, when we compare infants born by cesarean delivery as compared to vaginal delivery, we see that the father plays comparatively a stronger role in the infants that are born by cesarean. This makes sense also because so we did this study with collaborators in Finland, and there all the mothers that delivered their infants by cesarean, they got antibiotic prophylaxis to avoid infections. So this gets rid of their microbiomes, and it's then difficult for them to provide this seeding to the infant gut. So it's very good news to see that other individuals that are in close proximity can also substantially seed the infant gut. As a next step, we checked strain sharing between adult twins. This is, for us, an interesting scenario because these are adults that got their microbiomes probably similarly from their mothers, but then they are now, as adults, they are not living together anymore. So we saw that indeed strain sharing between these twins declined sharply with the years they were living apart, but still, and well, much strongly as compared to what they are missing from their mothers. So their age was not so important. What was very important is the years they live apart. So again, this is telling us that environment is playing a stronger role than what they are losing from what they acquire from their mothers at birth. We also assessed whether there was an effect of genetics, and we see that monozygotic shared more than dizygotic twins for each a category of years living apart. But this was not a very strong signal. So we see that genetics indeed seems to display a moderate effect for microbiome transmission. As a next step, we also assess transmission upon cohabitation. And there, what we see is that there are no differences among the people that live together for the gut microbiome. So the partners, the siblings, or them, or the parents with the children, they share all of them similar amount of strains. And they all share a lot more than people in different households, suggesting that indeed horizontal transmission more than genetics is playing a very strong role. And we went then one step further, as we saw that people in the same village within a population tended to share more than those in different villages. So indeed, we managed to reconstruct strain sharing networks. So in this plot, each dot is one individual sample colored by the dataset they belong to, and the connecting lines correspond to the number of strains they share. So we can see, for example, here that there is an island in Guinea-Bissau. These are quite separated from the rest of the populations. So indeed, strain sharing networks recapitulate host geography. We then went into exploring other microbiomes. We went for the oral one. As I said, we also included saliva samples. And we saw there that indeed for the oral microbiome, there was even more transmission than for the gut one. This makes sense as oral microorganisms have saliva as a transmission vehicle. And they are also aerotolerant, so they can survive outside our bodies. It will be easier for them to be transmitted. Indeed, you can see that people in the same household, so mother offspring, father offspring, or partners, they all share quite a lot, so up to 40%. And we also share very little, but something with people in the same population. This could be indeed through droplets with saliva being spread, or these bacteria staying in the environment and then being acquired by someone else. And on the mechanisms of transmission, it was very interesting to see that indeed, whereas for the gut microbiome, we see this strong inoculation from the mother, and then it declines. For the oral, it's the opposite. So the more you live with someone, the more you're going to share with them. So it's the older children that they share more, both with the mother and with the father, and also the fraction that is overlapping between both parents. So then, as a next step, we wanted to see whether all the species in the microbiome are transmitted equally, or if there is some that are more dispersal specialists, so are transmitted more frequently. Because this is something that we believe could have important implications for then the design of probiotics and other microbiome-based modulations. So this is just a formula on how we calculate transmissibility of a certain species based just on the number of transmission that we observe, divided by the potential transmission events that could happen. And we saw that indeed there is a lot of variation. Some bacteria are transmitted a lot more than others, but the rates of transmission are quite consistent across data sets. Meaning that one bacteria will be transmitted a lot or very little, regardless of whether it is in a population in Africa or in North America. But still, we can see that indeed this could be a property of the bacteria, and it's different bacteria that are more often transmitted vertically, horizontally, and also within population, so with these longer transmission paths. It was interesting to see that we have many under-characterized bacteria, so bacteria we know very little about, that are actually very commonly transmitted. And this was specifically, especially true for the oral microbiome. So there, the most transmitted bacteria, for several of them, they don't even have a name. For example, here we just see both for the genus and for the species, they just have a code, meaning that we really do not know much about them. We think that we should study this more in detail to see whether they can be relevant for our health. So we then assessed whether there are some phenotypical properties that could explain the transmission dynamics that we observe. And we saw that for the oral microbiome, we did not see strong associations. It seems as if there is something else that would explain why some bacteria are transmitted many more than others, as all of them basically can survive outside our body. So it's probably properties that help them colonize the new recipient. Whereas for the gut microbiome, it's mostly properties that let bacteria survive outside our bodies. So for example, gram-negative bacteria that, because of their cell walls, are more resistant, for example, to disinfectants and cleaning agents that we use in our houses, are more transmitted. And then for transmission across longer ranges, so people that do not live together, it's mostly aerotolerant spore-forming bacteria that are preferentially transmitted for the gut microbiome. As another step further, together with PhD students in the group, we analyzed whether another example of transmission was following the same patterns. So this is induced transmission. So through fecal microbiota transplantation. So this is used now as an approved therapy for infection with Clostridium difficile when antibiotics do not work. But it's been explored for many other conditions because we see that it works surprisingly well. So here we did just a meta-analysis of all the published studies on fecal microbiota transplantation, taking so the samples of the donor and the recipient pre and post transplantation. We saw, first of all, that strain engraftment, so colonization of the donor strains into the recipient gut, was linked to the clinical success. So indeed, FMT seems to work best when bacteria are actually colonizing. Also, a combined administration route and giving antibiotics is beneficial. So I think this is because we think from an ecological point of view, the antibiotics get rid of a lot of bacteria, so there is less colonization resistance, and then it's easier for the bacteria in the donor to be colonizing. But again, there were also some phenotypic properties of the bacteria that made them more prone to engraft. And we can see here again that the gram-positives tended to engraft less, so gram-negatives, again, as we saw for households, are engrafting preferentially. This is something that we still cannot fully understand, so but it's at least telling us that gram-negatives, besides maybe surviving better in the environment, they are also engrafting better. And then we can also see that there are some phylum-level differences. So for example, the firmicutes tend to engraft quite little, whereas others like actinobacteria or proteobacteria a lot more. As a next step, what we are moving now into is trying to link transmission of the microbiome to different diseases. Because we think that this is where, so after tracing the landscape of transmission in healthy individuals, this could actually be playing a role. We think that more socially connected individuals, so especially patients, it will be easier for them to recover their microbiomes. So if a person is more exposed to healthy organisms, then it's also easier for them to restore their diversity. As in many diseases, the main signature is actually reduced levels of reduced diversity overall. So indeed, I mean, even if there could be this worry of if you live with someone who's sick and with a dysbiotic, so disease-associated microbiome, as a healthy person, you could be acquiring these microorganisms, and this way the disease could be transmitted. We think, based on ecological principles, that it makes more sense that we have the opposite. So in this way, believe that it's as the healthy microbiome displays a higher colonization resistance, it will be more the patients that will benefit from exposure to healthy individuals. This is something that we are hypothesizing. We don't yet have the data to assess this, but we have studied so far two different conditions, two different papers. So one is neuroblastoma, which is a pediatric cancer, and we studied their patients with neuroblastoma together with samples from their mothers and their healthy siblings. And then we also had control families with mothers and healthy kids. And we saw again that infants with neuroblastoma had decreased microbiome diversity. Their microbiomes tended to be quite different from the healthy kids, whereas the siblings of the patients were similar to the healthy kids. So it seems that indeed it's not due to the family. Also, the mothers of children with neuroblastoma and the mothers with healthy kids had very, very similar microbiomes. So it seems that indeed this disease is also associated with alterations in the microbiome, both at the taxonomic and then also at the functional level. So the patients with neuroblastoma had more pathways for protein fermentation, whereas the healthy children had more glycoprotein and carbohydrate fermentation pathways in their microbiomes. But when we then went into assessing transmission, we saw that actually there were no differences. So the mothers were giving the same amount of bacteria, and also there were no significant differences in any specific bacterial species to the healthy or to the diseased children. So this suggests to us that all these alterations are likely to arise after the initial maternal inoculation. So we should not blame the mothers here. And microbiome transmission does not seem to, at least maternal microbiome transmission, does not seem to play a role. Another example was the study of children with autism, where we also analyzed samples from both the mother and the father. And there we did see that the children with autism were sharing less of their microbiomes, both with the mother and the father, as compared to their neurotypical siblings. And this was also correlated to their social communication skills based on validated questionnaires. So in this case, it seems that indeed children with children with autism, especially those that interact less with their families, get less of the health-associated bacteria from them. So there is indeed reduced family microbiome transmission there. So overall, just to start wrapping up, we see that actually we get our microbiome first from our mother when we are born, but then also proximate individuals are shaping this. So it looks like our social interaction are the main source of diversity for our microbiomes. Then we also get some bacteria from food, but it seems that food is shaping the composition of our microbiome overall. It's not really giving specific bacteria that are going to colonize in our gut. And we are also assessing transmission from other animals and the environment. But so far, and mostly for the gut microbiome, we do not see very strong contributions. So this is leading us to start thinking about how we can use what we learn from natural transmission to think of artificially induced processes to modulate our microbiome. So this could be probiotics, live biotherapeutics, or fecal microbiome transplantations, as I introduced. And we think that with more and more evidence, we can actually try to improve our microbiome with more targeted, more personalized approaches. So to conclude, we have seen that exploration, the relationship between the gut microbiome and mental health, is a field that is really rapidly advancing. Some years ago, when I started my PhD, there was evidence mostly only in rodents. Still, translational evidence is lagging behind, but it's moving fast, and we are getting there. And we believe it holds promise for therapeutic development, although still many questions remain open. Then also the metabolism of neuroactive compounds by members of the microbiome. We found it linked to both our physical and our mental health. So we think it's something that we should explore further. The tool is available to the public, and we are seeing that more and more people that have cohorts with patients without any mental health conditions are using this, and it helps with interpretation. And then that by improving the resolution of our strain tracking methods, this allows analyzing microbiome transmission. We see that mostly we acquire our microbiome as adults through our social interaction. So this has many implications, both for patients, but also to keep our health. The impact of disease is, as I showed, we have only two examples now, but we believe that more and more it's important to, now that we have enough evidence, when we design a study, include also a social network of someone into the study design to see where could this bacteria be coming from, especially the ones that are causing or sustaining the disease. So with this, I would like to finish my talk by thanking the labs where I've conducted part of the research and all my colleagues there, and then also the members of my team. So, thank you very much y estaré encantada de responder las preguntas.
Muchas gracias, Mireia. Por favor, levanten la mano y hagan su pregunta los que estamos en Zoom. Los que están en YouTube, por favor, escriban sus preguntas en el chat y acá las leemos. Adelante, Diego.
Am, Hola, muchísimas gracias por la plática. ¿Me escuchan?
Sí.
Va. Yo me quedé pensando que si bien las estructuras sociales explican la presencia o ausencia de algunas cepas entre individuos, Em, ¿qué más se sabe acerca de la abundancia relativa de estas cepas? ¿Realmente representan una amplia proporción en los microbiomas o están limitadas? Por ejemplo, representan al final de cuentas muy poco o representan mucho. ¿Qué es lo que se sabe?
Bueno, es una buena pregunta. Algo que también, si hablamos ya ayer, vemos que esto, claro, aquí, o sea, para cada especie tenemos una limitación metodológica que es que podemos reconstruir la cepa principal. Así que podemos aproximar a tu pregunta considerando que hay una cepa dominante para cada especie y es la que estamos traqueando para analizar transmisión. Si hacemos así, no tengo ahora los números exactos, pero sí vemos que no son ni o sea, no hay diferencias en que sean, o sea, una especie más abundante se transmita más, que esto sería un modelo de mass action, o sea, que si simplemente si una bacteria es más abundante en la población, pues será más probable que se transmita. Esto no es el caso, parece que realmente no hay asociación entre la abundancia de una especie o la prevalencia en la población y su transmisibilidad. Sobre esto, los números exactos, parece que sería una parte parecida a lo que vemos con presencia-ausencia, pero no tengo los números precisos ahora.
Bueno,
¿Alguien más?
gracias.
¿Alguien más? Ethan.
Hola. ¿Sí me escuchan?
Sí.
Yo tengo una pequeña pregunta acerca de bueno, como la herramienta que nos había mencionado, que es Metafian 4.
Mataflan, sí.
Gracias. Me acuerdo, creo que mencionamos ayer, pero no profundizamos está especializada en bacterias y arqueas, pero no maneja o no detecta el el genoma de los virus o todas estas cuestiones. ¿Qué impacto cree usted que podrían tener en todo lo que nos está mencionando de la microbiota intestinal y su relación con enfermedades mentales?
Es también una muy buena pregunta. En realidad, durante muchos años hablamos de microbioma, pero nos centramos sobre todo en el bacterioma. Y mis estudios son en bacterias y arqueas. Sí que cada vez hay más información y esto permite cada vez más estudiar la parte, o sea, el viroma, los viruses, y también los microeucariontes. Y ahora Metaflan, desde hace poco, un par de meses, también permite perfilar los viruses. O sea que desde ahora se puede hacer también. Y sí, hay algún estudio que relaciona, por ejemplo, el viroma con, con el estrés. O sea, parece que o sea, los viruses son menos abundantes que las bacterias, pero van a tener un papel importante. Y los eucariontes igual. Por ejemplo, hay blastocistes que es una especie que se consideraba como protista, que sería un patógeno. Y en realidad vemos que hay muchas personas que los tienen, son personas sanas, sobre todo los vegetarianos, que tienen un microbioma en general más diverso y en algunos casos más saludable. O sea que es algo que se está expandiendo y yo creo que van a tener también un papel importante, aunque todavía no lo hemos estudiado mucho.
Ok, muchas gracias.
Adelante, Luis.
Eh, Hola, Mireia, desde el punto de vista de los compuestos neuroactivos, ¿alguna información, dato que haya respecto a individuos dentro de todos estos análisis de datos, a aquellos que consumieron antibióticos? O sea, si hay evidencia epidemiológica respecto a alteraciones en síntomas neurológicos en asociación al consumo de algún antibiótico.
Gracias. También una buena pregunta. O sea, sí que hay datos, como he mencionado, que los antibióticos, evidentemente, aparte de eliminar las bacterias patógenas, también eliminan parte importante del microbioma asociado a nuestra salud. Pero evidencia, o sea, lo que vemos esto es una reducción de la diversidad y muchas bacterias consideradas buenas que no están allí. O sea que me esperaría que hay algunas de las funciones, como por ejemplo producción de butirato, que es importante y la vemos sobre todo cuando hay ya un ambiente anaeróbico con una comunidad bien establecida. Me esperaría que hubiera allí también afectaciones, pero no es algo que se haya estudiado en detalle.
Algo que se sepa respecto a la serotonina. Había escuchado algo respecto a cambios en los niveles de serotonina y la transmisión desde el intestino por el nervio vago hacia el sistema nervioso central y que pudieran, digamos, alterar de una manera en el corto plazo, pues, conductas, ¿no? Efectos directos en el comportamiento de las personas. Entonces, no sé, como quisiera saber si hay algo más como decirlo, contundente, que digamos que un tratamiento debería de prevenirse, por ejemplo, antibióticos frente a personas que están deprimidas o asuntos en ese sentido que pueden ser muy trascendentes en la calidad de vida de las personas.
Sí, es una buena pregunta y realmente el caso de la serotonina es interesante. Las bacterias vemos que la pueden producir por dos vías metabólicas diferentes, la que utilizamos los humanos y la que utilizan las plantas. Hay una más prevalente que la otra, pero sí están allí. Y en ratones sobre todo se ve que cuando dan antibióticos, pues la proporción de serotonina en el intestino disminuye muchísimo, demostrando que realmente no son solo nuestras neuronas y células humanas que tenemos del sistema nervioso entérico y conocidos que producen serotonina, sino que la serotonina intestinal es en gran parte producida por bacterias. O sea que esto sí que parece que realmente tienen en condiciones generales un papel muy importante y, como he dicho, esperaría que los antibióticos pues tuvieran efectos importantes. Creemos que en general se tendría que reducir todavía más la administración de antibióticos en cualquier caso por la emergencia de bacterias resistentes. Pero que yo sepa, no hay nada específico en estudiar serotonina en pacientes con depresión. Si encuentra algo, estoy encantada de verlo, pero
Okay.
creo que yo
Bueno,
sepa, no hay nada.
ok, muchas gracias.
Mireia, hay una pregunta en el chat. ¿La quieres leer tú o la leo yo?
Sí, claro, la puedo leer. A ver. Vale. ¿Cree que podría haber diferencias en el estudio de la relación entre microbioma y salud por la zona de donde se muestrea? El microbioma de zonas más altas del intestino podría dar más información. Pues es también una muy buena pregunta. En la mayoría de estudios se utilizan muestras fecales porque es lo más fácil de obtener, pero sí que hay estudios, sobre todo en modelos animales, donde se han estudiado varias zonas y vemos que hay una variación muy importante. Claro, lo que vemos en las heces es sobre todo el colon y allí es donde hay la mayoría de las bacterias, pero en la parte alta del intestino hay algunos estudios que se han hecho, por ejemplo, con vómito y sí que sé, claro, allí las proporciones son diferentes, pero como he dicho, se sabe todavía poco de la biogeografía de la microbiota en el intestino, porque claro, en humanos sobre todo, porque, claro, son muestras muy difíciles de obtener.
¿Alguna otra pregunta? Bueno, en lo que levanta la mano yo tengo una pregunta relacionada a dos. Sobre el caso de los niños con neuroblastoma, queda claro que la microbiota que les transmitió la madre fue la misma al hijo sano que al hijo con el tumor, pero claramente hay una disbiosis en el niño con el tumor.
Sí.
Mi pregunta es, ¿se sabe si esa disbiosis es resultado de los tratamientos que recibió o resultado del desbalance de la comunicación del cerebro con el sistema inmune en el intestino?
Sí, es algo que no mencioné, pero como era el primer estudio que se hacía en el microbioma neuroblastoma, recogimos muestras de los niños antes de iniciar cualquier tipo de tratamiento, o sea, treatment naive, para asegurarnos de esto, que no fueran, sí, también porque hay mucha variación de los tratamientos que se dan, es una mezcla de quimio, radio, con tratamientos biológicos, cirugía en algunos casos, pero las muestras que teníamos era esto, antes de empezar ningún tipo de tratamientos, o sea que sí que aquí sabemos que lo que se observa parece que es debido a la enfermedad. Podría, claro, lo que no hemos podido controlar al 100% es la dieta. Hablando con las familias nos dicen que los niños, o sea, el hermano sano y el que tiene el tumor comían de manera parecida, ellos les dan lo mismo, pero claro, y todavía no están hospitalizados, o sea que parece que no tendría que haber mucha variación, pero claro, siendo un estudio sí, de observación y sin... Sí, nosotros poder controlar todo, sí, podría haber también otras fuentes de variación.
Ok, gracias. ¿Alguna otra pregunta? Bueno, creo que tampoco ya no hay en el chat. Pues,
Perfecto.
pues muchas gracias por tu excelente presentación y por la clase de ayer también.
Gracias otra vez por la invitación. Encantada de estar allí.
Hasta luego, que estés muy bien,
Con
cuídate.
ustedes.
Vaya, hasta luego a
Gracias.
todos.
Muchas
Hasta
gracias.
luego,
Hasta
muchas
luego.
gracias.
Gracias.